Remember that protein is a polymer, is a macromolecules composed of the amino acid, which is a monomer. And then we look at the amino acid. There's the structure of amino acids, consists of two important functional groups. First one is the amino group, which is basic, and another one is a carboxyl group, which is acidic. Next, we have hydrogen atom attached to this. And the fourth one is the variable R group. This is the R group. There is the side chain. So this side chain, which is R, is important to determine the characteristics and properties of the amino acid. That's why we have 4 classes of amino acids. We're going to take a look at the classes after this. Okay, we look at the carbon is alpha carbon, which is asymmetrical alpha carbon. Because if you look at the mirror, it is not a mirror image when you look at the left and right side here. Okay, it's not a mirror image because this one consists of the amino group and this is the carboxyl. That's why the carbon is asymmetrical. These are the 4 types of amino acids or 4 classes of amino acid. Depends on the side chain. So the R here is just H, so it's non-polar. Okay, for this one, the R consists of the methanol, CH2OH. There's an hydroxyl, which is polar. So this characteristics of amino acids is polar. The third one, the R group or the side chain, okay, consists attached with the ammonium, okay, NH3 plus here. So ammonium is basic, contributes to the basic characteristics of the amino acid. And another class, R group or side chain, it consists of carboxyl, COO-, which contributes to the acidic properties of amino acids. So there are 4 classes or 4 types of amino acids. And another one, the properties, there are 2 jargon words. One is amphoteric, another one is zwitterians. Amphoteric means that the amino acids have both acidic and basic characteristics due to the amino group and carboxyl group. Amino group contributes to the basic characteristics and the carboxyl groups contribute to the acidic characteristics of amino acids. So these amino acids have both characteristics. Another one is zwitterians when it dissolves in water. So this is the structure of zwitterian, where the amino group changed to the ammonium ionic, NH3 plus here, ionic charged here, and carboxyl, COO-, so when it dissolves in water. So in water, it carries a positive charge and negative charged. This is the formation of peptide bond, removal of water. So then C will be linked to N and form the peptide bond. Okay, but take note, C here is from the carbonyl, C double bond O. So form the bond. Next, the levels. The level of protein. The first one, primary structure. This is just a straight or linear chain. Example, lysozyme consists of 129 amino acids. The secondary structure, fold structure, folding or coiling. The folds is known as beta pleated sheets and the coils is alpha helix. So this is the secondary structure for the second level of protein. Tertiary, due to interactions of R group or side chain. So this is the 3D conformational shape of the protein. So there's hundreds of proteins here. There's interactions, they're weak. There's strong interactions. For the weak interaction consists of the first one here. These are hydrophobic interactions and van der Waals. The second one, hydrogen bond. So there's an OH here with O. And then third weak interactions, ionic bond. NH3 plus are charged and O minus. So there's ionic. So these 3 weak interactions. I'm going to have another one which is strong interaction between sulfur and sulfur. And this is disulfide breach. So there are 4 interactions here for tertiary structure of the protein. And the last one, quaternary. Quaternary, aggregation of 2 or more polypeptide. In here example, there are 3 polypeptides. Polypeptides consist of amino acids. And when there are 2 or more polypeptides, okay, so this is quaternary structure. Example, collagen, there are 3 polypeptides. Another example, hemoglobin, 4 polypeptides. Each of the polypeptide consists of the heme component with an iron atom. So oxygen will bind to iron atom. So there are going to be 4 molecules of oxygen attached to the iron atom. And this hemoglobin, when it binds to the iron atom in the hemoglobin and will produce oxyhemoglobin. So there's a loading of oxygen here. Then we look at the types of protein. Just now the level of proteins, primary, secondary, tertiary, and quaternary. The types of protein, there are 2 types, fibrous protein and globular protein. Fibrous is insoluble. Okay, an example, collagen and silk, mainly composed of secondary structures. Another one is globular protein, it's spherical shape, and the dominant structures is tertiary and quaternary. Example, enzymes, proteins, receptor proteins, and myoglobin. So this is a figure that shows the types of protein. For fibrous, example is keratin, and for the globular protein, example is myoglobin. You can see the difference between myoglobin and hemoglobin. Myoglobin just consists of 1 polypeptide, but hemoglobin 4 polypeptides. So this is 1 polypeptide, and there's 1 heme component with an iron atom for the oxygen to be loaded here, right? Okay, next, denaturation. We have done with this denaturation as well. This is when the enzymes, example of the protein is enzymes, probably high temperature or high acidity. So this normal protein will lost its conformation or its shape, unravels, and the bonds will be broken, okay, at the active sites. So denaturation, so they're going to have this denatured protein, but from denatured, it can be converted to the normal protein through renaturation process.